Welcome back to the African Microbiomes and Health and Disease Symposium. My name is Anna Happel, and I look forward to chairing the next session, exploring Africa's ecological richness through its diversity. I think we're just putting up the slides. Yeah, so welcome back from lunch to the African Microbiomes and Health and Disease Symposium. My name is Anna Happel, and I look forward to chairing the next session, which is entitled Exploring Africa's Ecological Richness Through Its Diversity. I moved to South Africa 10 years ago and never left. And one of the reasons for that is that I fell in love with South Africa's diverse cultures and its people. Africa is one of the most biologically and culturally diverse continents on Earth. It has a vast range of cultures and languages and ecosystems, ranging from lush rainforests to dry deserts. Together with its genetic diversity of its people, this offers unparalleled opportunities for global health research. Its genetic diversity is mirrored by its vast microbiome diversity, providing opportunities to study how genes and microbiomes interact to influence health outcomes. Altogether, South Africa's microbial diversity offers both a resource and a driver for global health innovation. Africa's microbial diversity is not only a resource for the continent, but for the world. Genomics, environment, and lifestyle factors all influence microbial populations, which are understudied on our continent compared to other regions of the world. Every disease, every condition, and every region could benefit from this diversity. For example, we know that early life exposures to diverse microbiomes influence immune system development. Africa's varied microbiomes can provide insights into preventing autoimmune diseases, allergies, and other immune-related conditions that are becoming increasingly prevalent globally. Africa's microbiomes have a critical role to play to understand how microbial populations affect infectious diseases like TB, malaria, or HIV. Studying the interaction between microbes and pathogens that are relevant to our context can aid in the development of new treatments and vaccines with a global application. As increases in antibiotic resistance become a global concern, Africa's diverse microbiomes can be a source for unique microbial strains that can inspire novel antibiotics or treatment solutions. Africa's microbiomes can be harvested for microbial strains that can be included in the development of live biotherapeutics, for example, those targeted at improving sexual and reproductive health outcomes. And as diets and environmental exposures change, the prevalence of non-communicable diseases, such as cardiovascular disease, diabetes, or cancer, is increasing globally, including here in Africa. This provides an opportunity to study how microbiomes, diets, and environmental exposures influence disease risk. And as we prepare for future pandemics, Africa's rich ecosystems are a key area to be monitored for the emergence of potential pathogens. Every region should have the tools and technologies to monitor variants and to investigate pathogens in order to contribute to global efforts to prevent outbreaks and respond to pandemics. So what is needed to unlock the full potential of Africa's microbial diversity? Sequencing data will help to tap into the full potential of Africa's microbial diversity. Africa's microbiomes are vast, yet understudied, and sequencing data will be one of the necessary means to fully understand microbial function, diversity, and influence on human health. Professor Kwon has his academic home at the Reagan Institute in the United States, but he has conducted significant amounts of work here in the clinical site in Durban, South Africa, and he will share with us the value of African microbiome data for the world. Multi-omic data analysis approaches will play an important role to unlock the full potential of Africa's microbial diversity. Correlating metadata with genomics and transcriptomics and other omics datasets will allow us to gain a holistic understanding of microbial diversity and function, its interaction with the environment, the human host, and each other. Professor Scott Hazelhurst is a South African bioinformatician based at Wits University at Johannesburg, and he has contributed significantly to population and genomic studies on the African continent, and he will share some of his wisdom with us during his talk. And finally, Africa's rich ecosystems, like the soil or the ocean, can be harnessed for the discovery of new drugs, from antibiotics to anti-cancer compounds. Professor Marla Trindade from the University of the Western Cape here in Cape Town will reflect during her talk on the untapped potential of African microbial ecosystems for biodiscovery pipelines. So as I conclude this brief introduction, I would like to share an African proverb with you. Knowledge is like a baobab tree. No person can embrace it with two arms. And for those of you who don't know, baobab trees can live up to 5,000 years, reach a height of 30 meters and a circumference of 50 meters. And this is why this African proverb encompasses 3 key points I would like to advocate for. International and interdisciplinary collaboration. Africa's microbial diversity offers a platform for scientists, communities, and policymakers to work together to unlock its full potential. The generation of big data will be the cornerstone for understanding or for gaining a complete picture of Africa's microbial function and its relevance for human health. It has the potential to revolutionize health outcomes, leading to innovative solutions that are tailored towards Africa's unique challenges and opportunities. And I truly believe if we work together and generate data that allows us to address these gaps in global health knowledge, particularly how microbial diversity contributes to health disparities, we will be in a situation to decrease health inequities and improve outcomes for all. With that, I would like to hand the stage over to Professor Scott Hazelhurst. Then I will hand the stage over to Professor Marla Trindade, who is a DST NRF Saatchi Research Chair in Microbial Genomics and Director of the Institute of Microbial Biotechnology and Metagenomics, the University of the Western Cape in South Africa. She has established a microbial biodiscovery pipeline to develop and commercialize innovative products through the sustainable exploitation of indigenous biodiversity and from extreme environments for the application in several biotechnology sectors using genomics approaches. And her talk is entitled Ecosystems of Life, the Untapped Potential of African Microbial Diversity. Thank you, Marla.
Good afternoon, ladies and gentlemen. I just want to start by thanking Joanne and her team for this wonderful opportunity to just discuss some of the research that we've been doing at University of the Western Cape. As Anna mentioned, this is looking at the microbial diversity from a wide range of environments and harnessing their diversity and their adaptation, to their environments as a way to develop novel biotechnologies. And so for the last 15 years, which way do I point? There we go. For the last 15 years, we've accessed a wide range of environments from all over the world, looking at novel microbial diversity to develop biotechnologies that can be used in all those sectors. And those are all the projects I've been involved in the last 15 years. And this has the potential to contribute to a lot of the SDGs. I'm not going to take you through all of them. I'm just going to show you some of the examples just to illustrate just how untapped these resources are. And I'm going to focus on those that relate to health for the sake of this symposium. So as I said, we've accessed a large collection of environmental samples, metagenomic libraries, and cultured isolates. This represents a very large repository that we cannot exploit by ourselves. And so this led to the formalization of this collection becoming one of the DSI's biobanks in South Africa. So we're one of the core one of the 7 core biobanks in South Africa. We represent the microbial collection. We take samples, by the way, so if you'd like to submit to our collection, please do. We're also a Diplomics partner lab. Diplomics is another instrument funded by the DSI that does omics training. And so we're one of 8 nodes in South Africa that train staff and students at tertiary institutions on next generation sequencing. And so I brought these two together, and in collaboration now, we contribute 500 genomes to the 1K SA project. This is an initiative to sequence 1,000 endemic species of South Africa. This includes microorganisms, plants, animals, not humans. Siri does the humans. So we contribute to half of this. And through this partnership, what I've tried to do is that for each genome that we showcase internationally, for each culture that we showcase internationally, that we can provide full genome sequence as well as metabolic profiling of these strains. And so in this way, we're really trying to take a repository that we have and just make it available to as many people around the world as possible. So in my biodiscovery work, we use a wide range of techniques, both culture-based and culture-independent approaches as a way to access this microbial biodiversity towards developing biotechnologies. And the initial premise was that if we access environmental areas that are untapped or undiscovered, that this will lead to the discovery of novel microbial diversity, which then leads to novel biochemical and functional diversity that we can then develop new biotechnologies. And that has been both true and untrue. And I hope to share some examples of why that is through some of the work. So this is an example of taking a metagenomic approach. We identified a novel protein that when expressed in either 2 different bacteria hosts can produce a range of ornithine lipids. These are amphiphilic molecules. They reduce surface and interfacial tension. And what that means is they're great emulsifiers, and emulsifiers, chemical emulsifiers, are used in so many products and so many processes. You've probably come across more than 5 of them today. And so giving a more eco-friendly solution to these, to replacing these chemical surfactants. We've patented this process. We've got several patents on this because each of the congeners actually has its own properties. It can be either more or less soluble in water and oil. And the point of telling you this is that each one has its own specific properties that can make it applicable to a different product or a different process. So this has caused us to partner with a number of industries, both nationally and internationally, to see if we can get them into a commercial application. And so there's some examples with agriculture for droplet management in adjuvant formulations. This BioDX is a South African company. We're looking at using these compounds in treating biofilms in industrial wastewaters and cooling towers. We've got for fungal control on leather. With Cerebel, we're looking at call them LOLs, they're lysoornithine lipids, so in short, LOLs. These lysoornithine lipids form emulsions, nice creamy emulsions, so for apply and wash off lotions that we're developing. What else? Oh, and more recently, we're looking at whether we can include them in regulating metabolism and immunity. So this is very, very brand new work. I'm mentioning it here because it was the most relevant to the symposium. Aminolipids are very well-known signaling molecules in the human gut. And they're also activators of G protein-coupled receptors. And since these receptors are some of the most common targets for therapeutic development, and also these aminolipids regulating these receptors that affect so many different diseases, they are currently getting more attention as possible therapeutics. Also, just last year, not my work, but gut bacteria were found to be the main producers of ornithine lipids. This is the Akkermansia mucinifera bacteria, and they regulate inflammation in the human gut. And so, again, this is a very good reason for us to now be moving into this area. And so we've just initiated a project with Corinne Smith at Stellenbosch University, where we're looking at whether our recombinantly produced ornithine lipids can have any effect on this. And so we've used the zebrafish zebrafish model for intestinal inflammation. And there's good signs already that one of our congeners is able to have a good safety profile and also regulate epithelial function. So there's good signs that, and I'm really sad Prof. Ntuzi isn't here, because we currently have some grants being reviewed, and I just wanted a nudge, wink, wink, so that we can expand on this work. Right, so I hope that by now I've impressed on you how important biosurfactants are and why they are this century's one of the most multifunctional molecules of the century. And so we're really putting a lot of energy into diversifying the compounds that we can produce for all these range of applications. And so we're doing a wide range of expression strain development. This is either through enzyme engineering or using different platform organisms, to really expand what we can provide to industry. We're also looking at other classes of biosurfactants, not just these aminolipids. So lipopeptides is another class that we're interested in. But one of the largest research programs in my group at the moment focuses on the drug discovery from marine microbiomes. We have, through several projects, established a very large collection, as I mentioned at the start, where we've screened for these organisms from very unique marine invertebrate species that we've harvested off our coastline. And hundreds of them have very good activities against multi-drug resistant strains, anti-cancer, anti-inflammation, all sorts of activities. And so we use top-down bioassay guided isolation or genome-guided, bottom-up genome-guided approaches also to identify these novel compounds and see if we can push them into the drug discovery pipeline. So just as an example, preliminary photomotor response activities that we generated against zebrafish embryos enabled us to establish a collaboration with a Mauritian preclinical drug company, Axonova. They're specialists in screening and developing drug leads towards treating neurodegenerative diseases, such as Alzheimer's and Parkinson's. So we currently have several of our leads in their pipeline at the moment, which is really exciting. But in an effort so this is actually the work that I like to do, that I was trained to do here at UCT, by the way. I want to match the bioactivities that these strains produce together with identify the pathways that are responsible, the biosynthetic gene clusters that are responsible for producing these activities. And so to do this, we apply genome sequencing to all our strains. And to date, we've sequenced almost 400. Just on the slide is just a subset of these. So just over 100 of these strains. We've identified new species, new genera, but the most exciting part is just looking at the secondary metabolite capacity that is harbored by these strains. For the classes of compounds that we're most interested in, these are typically the ones that are associated with some kind of pharmaceutical application. We see that only 6% of these have been characterized. What that means is that over 90% of these pathways represent compounds that have yet to be discovered, yet to be characterized, and this is even from species within very well-studied genera. So there is just so much exciting work happening here. And what we also find is that in a lot of our examples, we have species that actually look phylogenetically the same. And when we do pan-genome analyses, we realize that to a quarter of their genomes are actually accessory regions, representing biosynthetic clusters that are unique to that particular strain. And so this is a very good message for microbiome work where, amplicon-based studies, I know I'm preaching to the choir here, but these are no longer relevant for wanting to learn about function and how these organisms, what they're doing and what they're capable of doing. And yes, and let's not overlook even well-studied organisms. There's a lot still to be discovered. So one of the major bottlenecks associated with this kind of work, trying to produce these compounds from the native organisms, is that they don't like to produce them in the laboratory. And this is due to very tightly regulated networks. Also, they produce them produce very small amounts of these activities. And this has got to do with their natural function. Secondary metabolites by bacteria is to give them a particular advantage in the environment, in response to certain environmental conditions. And so they don't need to produce this constitutively all the time. This does make it very difficult to move it along a biodiscovery pipeline. And so our chosen approach is to take a heterologous approach, all sorts of reasons for why that has advantages. And so we focus on a number of classes of compounds, the lantipeptides, non-ribosomal peptides. I said earlier why we're interested in lipopeptides as biosurfactants. We've got carotenoids, EPS, and also the signaling molecules. So we do look at regulatory in these non-model organisms so that we can better understand and also better engineer future strains for their production. One of the examples of this is lantipeptides. So this is my baby. I have two human babies, but this is my other baby. This is Thalassomonas viridans. It's a bacterium. It's a strict endosymbiont with marine invertebrates. This particular strain was isolated from oysters. And the reason I love it is because it's got this ginormous genome and is just jam-packed full of really exciting novel things to look at. Included were class 1 and class 2 lantipeptides, which we could not detect to be produced by the native organism. So we engineered very nice expression systems where we have the post-translational modification machinery and also fusions to better solubilize and purify these peptides. And we were able to show 2 of those 3 are produced in a heterologous host. And again, demonstrates the benefit of taking genome-guided approaches. You can actually start to discover things that you wouldn't be able to otherwise. To investigate the various forms of the peptides, we did do bioactivity assessments. We tested against a range of escape pathogens and other bacteria and yeasts and got no activity, which was very depressing. But this actually is not unknown for lantipeptides from gram-negatives. There's actually just a handful that have been characterized, unlike the gram-positive counterparts. And none of the characterized ones have antimicrobial activity. And actually the biological role currently remains unknown. So we had the ability to screen in zebrafish platforms again. And to our absolute astonishment, our peptide, one of our isomers, one peptide's isomers has sedative effects. It's producing similar profiles to diazepam. And then the second peptide has the complete opposite. It has stimulatory effect. And when you combine them, you actually abolish the activity altogether. So please don't ask me questions about this. This is very early work. We don't, I can't explain it. I don't know how it works. But what it does demonstrate, this was, as far as we know, the first study that utilized zebrafish platforms for screening for ribosomal peptides. And so it just demonstrates that there's a lot of work that we can still do towards discovering compounds from microorganisms for treating a range of diseases. So to close off, clearly we know the power of microbiomes, and this is either studied as individuals, isolates, or as communities, offer enormous discovery opportunities that can impact many different aspects of life on Earth. And there's still so much that we don't know. There's still so much to discover and describe. And, this is going to keep biologists, not just microbiologists. It's a very exciting time to be a scientist because there's just so many tools available and so many technologies that we can access and so much for us to discover. So it'll keep us busy for a long time. And this, little word of warning, as alluring as genome-guided discoveries are, there are caveats, and it's important to really do the functional analysis and investigation of what these organisms do and how these pathways work. So on that note, I have lots of people to thank. So lots of collaborators, lots of researchers that have contributed to some of the work that I've presented here, and obviously a lot of the funders that have assisted us in this work. Thank you.
Thanks so much. Okay, I think I will invite now Professor Scott Hazelhurst to the podium. He is a professor of bioinformatics at the School of Electrical Information Engineering and a senior scientist at the Sydney Brenner Institute for Molecular Bioscience at Wits University in Johannesburg, South Africa. His research focuses on bioinformatics with a particular interest in genomics and data science. Professor Hazelhurst is a collaborator on the H3 Africa Wits in-depth partnership for genomic studies project, and he's co-leading the Microbiome project within this project. He has contributed to several other H3 Africa initiatives, including those studying population diversity. He is PI on the DSI Africa Madiba Research Hub, which investigates multimorbidity in Africa, and his talk is entitled Roots of Health, Applying the Ecological Wisdom in African Human Genomes. Thanks.
Thank you. I was really honored to get the invitation to come and speak, particularly as it's an outlier talk about human diversity. And then a few weeks after the invitation came, I thought maybe it was mistaken identity, because when Joanne sent me the proposed title, I didn't know anything about ecological wisdom at all. And in fact, when I saw Samir this morning, he expressed surprise that I'd been asked to talk about something so philosophical. But I take the challenge that the best way of learning about something is to try to teach it. So I'll talk about that. So one of the things we do with human genomics is to explore human history, and we do a lot of this work in Africa. It's an area that is very controversial and sensitive. And so I thought I would start by a cautionary tale that shows complete lack of ecological wisdom before I try and give a definition for ecological wisdom. So the cautionary tale comes from a cognate discipline that also looks at human and pre-human origins, and that is in paleontology. Maybe I'll just try and mime. Great, thank you. Great, thanks. I can see it. Yes, it helps my miming.
Thanks for the patience, Scott.
That's right.
Viewers, you're trying to be as...
Yes, I understand. Right. So comes from paleontology. So at WITS, we are celebrating this year the finding of the Taung child skull. For those of you who don't know, I think this is one of the major scientific discoveries of the 20th century, because it gave a completely new insight into the origin of pre-humans, and which led to later work that showed the origin of humans was Africa. Now, despite this work being published remarkably quickly in Nature a few months after the finding of the skull, it was really roundly rejected by the establishment European and North American scientific community, because partly because they were taken in by that hoax on the right-hand side, the Piltdown Man. But the underlying problem was really the preconceptions of the community at the time, which said that obviously human origin was probably a few hundred yards from the British Museum. And so the idea that these hominins, it's a 2.5 million year old skull, so it's pre-hominin, it means pre-human, but it's hominin, could have come from Africa was just not something. In fact, it was called preposterous by Sir Arthur Keith, who was the leading paleontologist of the time. And I think that shows the danger of of preconceptions. We'd like to think we're better than those people. I'm sure we are, but I think we all suffer from those preconceptions. I think it's a good cautionary tale to bear in mind. So we now run the risk that moving on from the paleontological debate, where we're saying, well, is it Africa or is it London? To maybe, if we look at the human reference genome, we will say, well, it's probably the controversy is, if you look at the human reference genome, it's obviously the origins of human is a few hundred meters from Cambridge. And the only question is it Cambridge, England, or Cambridge, Massachusetts? So that may be a bit of a cheap shot, because I think most people involved in the Human Genome Project are well aware of the shortcomings of the reference genome and the need for there to be a much more diverse representation. But nevertheless, that's where we are. And I think that's perhaps a good place to talk about what ecological wisdom is. So I've done a fair amount of reading, and in the short time I've got, this is the best definition that I could find for a minute definition. So it's really about a process. It's about emphasizing understanding and living in harmony, approach to environmental ethics and sustainability that draws on both scientific knowledge and traditional scientific knowledge. And there's a set of values that go with it. This comes from the same paper where the definition is. So what I'm going to try and do is interpret this definition, not literally, but metaphorically. And I see these values as the values that we could strive for when we look at human genomic diversity and perhaps diversity more generally. So let's look at genomic diversity in Africa. And I'm going to start by actually showing a map of the linguistic map of Africa as a crude proxy for genetic diversity. So here we have the major linguistic groups: Afroasiatic in the north in blue, the Nilo-Saharan in yellow, the Niger-Congo in red and orange, and then the other population groups, and in particular I want to show the Khoisan, which the range is shown in green. So the Niger-Congo group's divided into two. Group B, which is shown in orange, is better known as the Bantu-speaking languages. That, to put it in perspective, Bantu-speaking languages are about as old as the Indo-European language groups, and there are as many Bantu languages as there are Indo-European languages. If we go back 12 years, the only major whole genome sequence resources with African data in were from 1,000 genomes, and they're shown in those white circles. There was one resource from a Bantu-speaking language, and the other four came from the Niger-Congo A group, two in Nigeria, and two further to the west. And the rest of Africa had not been sampled at all. Now, this is not just symbolic. As important as symbolism is, it comes with real consequences. And I'll take pharmacogenomics as a case study, because I think it's fair to say that generally drugs are not designed with Africans in mind, and certainly not designed by Africans. And that's a huge problem. And particularly as we know that genetic diversity affects both efficacy and safety of drugs. So let's take two examples. The first is efavirenz, which is a key frontline HIV ARV. It's being phased out in some parts of the continent, but still very important. And there's a whole stream of research. My colleague Colin Massimarembwa and Colette Dandara from UCT were involved in much of this work that shows that variation in the CYP2B6 gene is very important for drug safety of efavirenz, and that in certain parts of Africa, like Zimbabwe, the variant that is common meant that there was very significant adverse drug reaction to efavirenz at the recommended World Health Organization dosage. And by reducing the dosage by a third, you could get the same therapeutic effect at much reduced adverse drug reaction. And that unfortunately came far too late in the day. So it's very problematic of this understanding that one dosage fits all. So what's true for Europe is not going to be true in Africa. And in fact, we need to be much more nuanced in our approach. So we have to look at diversity within Africa as well. My other example is G6PD, which is a gene on the X chromosome. So this is some work that we did. In particular, we explored an area in Mpumalanga called Bushbuck Ridge. And what we showed is that in Bushbuck Ridge, among men, depending on your ethno-linguistic background, you had a very different probability of having a mutation which gave you G6PD deficiency. So G6PD deficiency is serious because you can have adverse drug reactions to a whole series of drugs that are shown over there. So if you are in a particular group, I'll call it group A. If you're a man, you've got a 16% probability of having G6PD deficiency. If you're in group B, then it's probably a non-existent chance of having a G6PD deficiency. So that shows that we need to be much more nuanced, and we often see, unfortunately, in the scientific literature, that there is this flattening, that Africa is described as a single place, that all Africans are the same. Clearly false. Genetic diversity, social diversity needs to be taken into account. Now, people in the human genomics community have not just sat around moaning and whinging about this, they've done things about it. And I'm going to talk about the success of the H3 Africa program, which was established about 12 years ago by African scientists with strong support by the Wellcome Trust and the NIH. So H3 Africa was a collaboration of many research groups. Most of the work they did was autonomous. They all had their own research goals, but they also worked together, and I think were very successful in changing the landscape of knowledge about human genomics in Africa. So one of the projects that H3 Africa did as an example was building common resources. So going out and doing a large-scale sequencing of underrepresented groups in Africa to bring in extra genomic diversity. And these resources are still being used today. And this led to some significant work, including work looking at the consequences of genetic diversity, using this to explore African history and understanding the medical consequences of that diversity. So that was something that was done collaboratively, but all the different research groups of H3 Africa had their own individual research programs, and there were well over 100 papers produced by the H3 Africa group in a very short period of time, exploring a range of different problems, disease, building resources, and building capacity. So I think it's an example where some of the values that are expressed in ecological wisdom were shown to be successful. One of the strengths of H3 Africa was the focus on the communities and the participants in which the groups worked. So the idea of engaging with communities before research happened, engaging with local research partners, and then giving feedback to participants and communities of the results of the research was seen to be very important. So having been through the process, I think it's an example of a successful research project that incorporates many aspects of ecological wisdom. Now, I'm not saying it was perfect. Certainly, people are people. They come with their own agendas. There were many acts of a commission and a mission. We could do things better. I'm sure we could do things better if we did it again. But nevertheless, it's an example of where Africans could do things. Africans could do work in Africa of great significance. And I think it's put us in a place where human genomics work can be done much more effectively in Africa. So whereas 10 years ago, we looked at the task of going out and sequencing 350 individuals with high coverage sequencing and doing all the bioinformatics work here as a major undertaking. And it was a major undertaking. We now have serious scientists who are talking about sequencing millions of human genomes. And there are existing projects that are now looking at thousands and tens of thousands of human genomic sequences. And I think this is possible. The whole cycle from sample collection, DNA extraction, sequencing, bioinformatics analysis can be now done on the continent. So I want to come back to the slide of ecological wisdom. And I think of understanding the relationship between our pursuit of knowledge, of science, and I'll call it wisdom because that should be in the title of was in the title of my talk, and the relationship between that and the scientists and the communities in which we work. So I think those values allow us to do better science and to do science better. But I think also we have to recognize is that there can be tensions. So I think, for example, we have to be honest about saying is that sometimes there's a tension between those values and doing things efficiently in the short term. Sometimes it's easier to outsource the work to someone to get it done quickly rather than, for example, looking at integrating knowledge or looking at justice and equity. There are also very human tensions. So if I want to promote my own research group, I want to get a particular grant, I want to advance my career, that's going to be in tension with some of those values. So I think we have to be honest. Yes, that allows us to do better things, but we have to be very conscious about adopting these values. It doesn't always come naturally. So I think I'm going to stop there, other than to say, okay, well, I had a list of thanks and thanks to all the funders, but I'll mind that. Thank you.
Thanks so much for a very inspiring talk. And yeah, our last speaker of the session is Professor Kwon, who is a physician scientist and director of clinical operations at the Reagan Institute of MGH, MIT, and Harvard in the United States. Through this work, Professor Kwon aims to advance scientific knowledge in immunology and to contribute to the development of new strategies for combating reproductive conditions like bacterial vaginosis. His group has contributed one of the largest data sets of South African vaginal microbial sequences to global databases, allowing novel insights into this condition, which are relevant to improving treatment options. And his talk is entitled The Ecological Value of African Microbiome Data for the World. Thanks so much.
Great, thank you. It's a real pleasure to be here in Cape Town. So, as was sort of discussed earlier today, we now have a greater understanding of just how much vaginal microbiota impact a number of reproductive health outcomes. And particularly, we sort of came at this by first wanting to understand how they might impact HIV acquisition risk. And in part, as was framed earlier, this is because, like many of these adverse reproductive health outcomes for women, the largest burden occurs within young women within sub-Saharan Africa. And this is certainly true for HIV. And so, in part motivated by this, we helped to sort of start a study along with collaborators at the Nelson Mandela School of Medicine in Durban called the FRESH study. And FRESH stands for Females Rising Through Education, Support, and Health. And this study has, over the past 12 years, enrolled over 3,200 young women, all HIV uninfected, within a community in Umlazi, just outside of Durban, South Africa. The women come and they actually get tested for HIV twice a week, just through a finger-prick viral load test. They also have quarterly pelvic exams where they get tested for sexually transmitted infections and have Pap smears. And one of the things we're very proud of is that they come not just to participate in research, but to also be involved in a very intensive poverty alleviation and empowerment program where they spend 3 hours each visit there over the course of 10 months. And most of the graduates, after completing the program, go on to either a job or to finish schooling or to an internship. And yet, this actually doesn't really impact the HIV acquisition rate. So the rates remain just as high as in the community and just enormously high within this population. So we really wanted to understand, well, what are some of the biological factors that we could better understand to sort of try to create and inform the development of interventions that might be able to prevent HIV within young women in sub-Saharan Africa. And one of the first things that we looked at was the vaginal microbiome. And sort of has been mentioned before, what we saw was very different than what we might actually see within young women of reproductive age in the US, where 90% of women who are white in the US would have a lactobacillus dominant vaginal microbiome, whereas in this cohort, it was only about a third of women who had that kind of microbiome. Most of the women actually had microbiomes that were comprised of diverse anaerobic bacteria. And this actually, we then demonstrated that these women with these more diverse vaginal microbiota had higher levels of inflammatory cytokines within genital secretions, had more activated CD4 T cells within the cervix, which, because it's the first cell thought to be infected by HIV, might confer an increased biological risk for acquiring HIV. And what we saw prospectively was, in fact, that women with these more diverse vaginal microbiota actually acquired HIV at over fourfold higher rates than women who had a microbiome that was dominated by a single species of Lactobacillus crispatus. And as Sharon mentioned earlier, we now have an appreciation that these vaginal microbiota have an impact not just on HIV, but on a number of really important adverse health outcomes, such as HPV, cervical dysplasia, cervical cancer, preterm birth, and fertility. But to really get more to sort of mechanistic understanding, we need to actually have good reference databases to understand these bacteria. And one of the problems that's existed in the field is that when we first started this project a few years ago, if you looked at the reference databases, there were actually very few bacteria that were from the female genital tract. Most of them were from the gut, and also very few example isolates. So one of the things that we've done is we've sought to expand the example genomes and metagenomes out there by taking concerted culturing efforts, particularly of South African isolates, and providing whole genome sequences of those, combined with other efforts in the North America to expand those data sets, as well as contribute metagenomes from fresh here to sort of vastly expand our understanding of what are the metagenomes and genomes of vaginal bacteria from particularly African women. And that's kind of led to this database, which we called FemCat, which is the Female Genital Tract Metagenomic Catalog, which has now over 26,000 genomes split into these species genome bins, or SGBs, which basically comprise 605 different SGBs with contributions, again, from reference databases, isolates that were contributed by us and also sequenced by others, and also these metagenomic assembled genomes. And these 605 SGBs include about 500 that were known to exist in the female genital tract, but also about 100 unique SGBs. And we really tried to be thoughtful about making sure that there was good representation of both North American and African isolates across relevant species within the female genital tract. And in doing so, we've actually greatly expanded the pangenome, or the collection of genes associated with these different bacterial species, both known to exist in North America and in South Africa. So, and there's been some interesting insights about the uniqueness of isolates from different geographies. For example, if you look at Prevotella bivia, which is an important anaerobe within the female genital tract, you can see that isolates seem to be an offshoot of South African isolates. And if you look at sort of some important antibiotic resistance elements, you can see that there are these interesting islands with the ERMD gene, which confers antibiotic resistance to a variety of different antibiotics, such as azithromycin, clindamycin, and erythromycin, and that these exist specifically within the North American isolates. And this was confirmed phenotypically as well. So there are interesting sort of phylogenetic structures within some of these relevant species within the female genital tract. Also, there are really interesting relationships with genital inflammation. So if you look at these different SGBs, even within specific species, you can see different patterns of inflammatory cytokines associated with different SGBs within these species. And in general, you can see this clustering of different SGBs with different patterns of inflammatory responses. And interestingly, within these SGBs within a particular species, you can see unique relationships with inflammatory cytokines. But we are also interested in looking sort of like what are some of the drivers of the diversity within this particular species? And could we understand strain interactions? And so we took sort of the genomes of all the isolate strains that we had from both North America and from South Africa. And then we sort of, looked at the diversity of all of those strains, and then we selected specific isolates, sort of marked in yellow, to select 100 strains of Lactobacillus crispatus that sort of represented the global diversity of Lactobacillus crispatus genomes that we had. And then we took those isolates and then we grew them, and then we collected supernatants from them. We filtered it, and then we did an all-by-all assay where we looked to sort of see is there anything in the supernatant of any particular Lactobacillus crispatus that might actually have an inhibitory effect on the growth of another strain of Lactobacillus crispatus? And could we use that methodology to kind of learn some of the strain-strain interactions that may be occurring that shape subspecies ecological diversity? And what we saw was that there were certain clusters of killer strains that were able to kill certain clusters of target strains. But this was not uniform. I mean, this was not universally true, that they basically were really specific for a certain set of these subspecies. And this sort of all-by-all interaction of 100 strains by 100 strains included over 10,000 different interactions. And from that, there are 25 killer strains identified and 32 sensitive strains identified. And then, so we were wondering, okay, well, what is the mediator of this killing? So we used a biochemical approach where we took these supernatants and then we basically used different protein fractionation methods to try to identify where the activity was being localized by doing sort of chromatography, as well as different types of chromatography, until we could actually get to a single band on a gel. And so we sequenced that band and identified that it was being mediated by a novel bacteriocin that was being secreted by Lactobacillus crispatus. And based upon the amino acid sequence, its nearest neighbor was another bacteriocin that had been described in Lactobacillus helveticus, but it was only 45% related to that nearest neighbor. So it was a pretty unique bacteriocin. And it seemed to have its activity through affecting sort of cell wall integrity. So this bacteriocin was lysing other specific sensitive Lactobacillus crispatus strains. And if you looked at sort of the overall pattern of killer strains and sensitive strains, what you could see is that these killer strains sort of clustered, and they killed sensitive strains that were phylogenetically distant from them. So they weren't killing the most closely related strains around them, but rather things that were more distant. And interestingly, when we looked at the geographic distribution of these killer strains, you can see that actually most of them occurred from South African isolates. And most of the strains that they were actually killing that were sensitive were from North America. So it provides some insight into that's not in any way a statement beyond what it is. But this provided real insight into sort of some of the unique properties of strains from different geographies and how they might be important. You know, we're also interested in looking to sort of see, because again, there are these very different sort of baseline proportions of different microbiotic communities, how they might be affected by an LBP containing Lactobacillus crispatus. Because ultimately, you know, even if we don't understand why these communities, understand the drivers of these community composition, we think that being able to achieve a Lactobacillus crispatus dominant community is more optimal for these health outcomes. And so we did some analysis from this trial that was done in the US of Lactin-V. So Lactin-V is LBP, a live biotherapeutic product, that consists of a single strain of Lactobacillus crispatus called CTV05. This was actually isolated from a healthy donor in Seattle in the US. It's administered intravaginally as a freeze-dried powder at 2 × 10⁹ CFU per dose. And this was a placebo-controlled trial that was done in the US. And it showed a moderate effect on recurrent BV as the clinical outcome. So those participants who received the Lactin-V product had a 30% rate of recurrent BV at 12 weeks, which is because the product was dosed for 11 weeks, was just after the product was stopped versus placebo. And at 24 weeks, there was still a modest effect. So, but this original paper had only looked at clinical outcomes using Amsel and Nugent score. So we'd actually been doing some work to study sort of the molecular outcomes. And also because we thought it was also important to understand how this product might be working in women in sub-Saharan Africa, we conducted a study of Lactin-V in the FRESH trial, in the FRESH clinic in South Africa. Now, there's some important differences in the way that these two studies were conducted. So the US Lactin-V trial involved 228 participants for randomized 2-to-1 to intervention versus placebo. Fresh Lactin-V was much more modest at 44 participants. women in the US study received vaginal metronidazole, whereas in South Africa, we gave oral metronidazole, which is the standard of care. And then women in the US received 11 weeks of dosing of lactin V or CTV05, whereas in South Africa, we gave it for 5 weeks. Now, given those caveats, though, we still sort of had outcomes measured immediately after dosing cessation, which was 12 weeks in the case of the US study and 4 weeks in the case of the fresh lactin V study. And then we also had follow-ups at 12 weeks after. So if you look at baseline, you can see that women, in fact, in both studies had these diverse anaerobe dominated communities. After treatment with metronidazole, they both were shifted to a lactobacillus annuris dominant community, which has been reported in multiple different studies looking at BV treatment. Interestingly, if you looked immediately after product cessation, you saw that there was a response. So orange here represents Lactobacillus crispatus, and you can see that a good number of the participants in both the US and the fresh lactin V studies had a response and established a Lactobacillus crispatus dominant community. But one of the interesting differences was 12 weeks after stopping the product. This was retained in participants in the US, but was actually lost in participants in the fresh study. And then these were some intermediate time points that were sort of unique to the two studies. So looking actually at the response or the benefit ratio, what you could see is that, again, immediately after stopping delivery of lactin V, you could see that 28% of participants established a Lactobacillus crispatus dominant community in the US, whereas 41% actually established a Lactobacillus crispatus dominant community in fresh. So both populations seem to have a significant response to lactin V, but 12 weeks after, you seem to maintain that in the US. But within fresh, 12 weeks after stopping, you actually lost it, and there was actually no difference between those participants in the lactin V arm versus placebo. So there does seem to be differences geographically. Now, this may be for a number of reasons. Obviously, there were some important differences in the way that the study was conducted. There are also very different baseline distributions of these bacterial communities within these different populations. And so, we want to better understand what were the drivers of these differences and sort of the durability of response, even though both were able to sort of establish these lactobacillus dominant communities with treatment. So these are just some of examples of, the way in which we've tried to, with, a lot of partners here in South Africa, contribute additional genomes from the female genital tract, and particularly from female genital tract isolates from women in sub-Saharan Africa and South Africa. And also, some of the potential implications and how it may sort of expand our understanding of, what are the mechanisms by which these vaginal microbiota contribute to disease, but also how we might be able to best intervene. So I just want to kind of thank, obviously, a lot of different partners, but particularly a lot of people in this room who contributed to the work that we've been done here in partnership. We've been kind of I've been working here in South Africa since 2007, and have always sort of really valued my relationships with all the partners here. So thank you very much.
Yeah, thanks so much for 3 inspiring talks. I'll invite the speakers to sit down in these comfortable chairs, and I'll invite the audience here in the room. I'll get you a blanket. I'll invite the audience here in the room and online participants to share any questions they might have. Janneke?
Thank you all very.
Is it working? Yes. For very inspiring talks. Thank you. I have a question for Doug. Very nice presentation. Thank you. Really nice work. I noticed when in your sequencing data that some of the women after the metronidazole, they had a lot of Gardnerella left over. Just quickly glancing, when you zoom into those women, are they the ones who failed the colonization, or do you see differences in? Because the Gardnerella, I think, may play a special role there.
Yeah, so for the US lectin-V study, that seems to be true. Like if you actually have more lactobacillus after treatment with metronidazole, you have a higher probability of colonizing with Lactobacillus crispata. So reduction of those anaerobes and sort of replacement with any lacto, including Lactobacillus inter, seems to sort of be a favorable predictor for successful colonization of Lactobacillus crispata. We don't know in the fresh group, in part because the numbers are smaller, but also because we haven't completed that analysis.
Okay, and also within the anaerobes, I think some may be worse than others, maybe.
Yes, whether it's Gardnerella specifically versus just the absence of a lactobacillus, we're not sure because the numbers get very small at that point.
Unfortunately,
so, yeah, right.
Thank you,
Heather.
Thanks very much, everyone. But I have a question for Doug too. I was wondering, Doug, why you chose 4 weeks only of the LBP, and if you think that rather than geography may have partly contributed to the differences. Okay.
Yeah. I mean, in part, it was a balance between sort of practical aspects and also, I mean, I think everyone recognized with the US Lactin-V study that an 11-week intervention is not ideal. So the question is, could we actually significantly shorten that and still see success? I mean, to me, what's interesting is that you actually do establish, the same, if not more, Lactobacillus crispata dominance within that study, but the durability is dramatically less. And whether or not that has to do with the length of time, it's quite possible. But we just don't know yet. So we have to better understand that.
Sarah has a question, and then Dash, please.
I will say, like, one of the ways we're looking at those that we actually have host cells. So we have cytobrushes from that study, which we're doing transcriptional analysis. So we want to sort of see if if it perhaps takes longer for there to be transcriptional changes, to then sort of perhaps promote durability. So that's one of the ways we're kind of looking at that.
Okay, thank you all. This is Sarah Gitome from Kemri. Thank you for the thought-provoking presentations. My question too is for Doug, sorry. So you talked about lactobacilli strains, some killing off others. And I wondered whether you've also looked at maybe presence of other infections like STIs or even candidiasis, and whether that has an effect on the longevity of the lactobacilli strains.
Well, we have STI data and also Candida data, or at least we can see yeast forms that have been called on like Nugent scores. We also have some 18S data where we've sequenced some fungi. There's no obvious relationship that we sort of see now, but that doesn't mean that it doesn't exist. I think we just haven't looked as deeply as we could there. So, but I think it's a great thought.
Hi, can you hear me?
Yes.
So I'm going to break the Doug Kwon spell and move a question to Prof. Hazelhurst. Thank you very much. All your talks were very interesting. I just wanted to know, in terms of the pharmacogenomics and the studies that you did in the Africans, whether you looked at any epigenetic effect regarding inflammation and either the expression of those enzymes or the drug transporters that affected, for example, efavirenz.
So the efavirenz work was not my personal work. It was work that was done by Colin and many of his collaborators over time. And I think I don't think there was epigenetic work, but they did a lot of other lab work working with the individuals. And they did some of the work that you asked about, but I'm not familiar with the details of it.
Oh, okay, thanks.
Thanks. Hi, it's Nikki Tiffin from UWC. So I've got a question about the non-human material, so microbiome material and I guess microbial data and samples in general. That is, I'm wondering what the relevance is when you move these samples off continent or out of your countries. What is the implication with the Nagoya Protocol and CBD? And especially when you're developing products from resources that originated in particular African countries, how you manage the governance and legality of that? Thanks.
Hi, Nikki. Thanks. So that's work that keeps one of my staff busy a lot. Is just the compliance and applying for all the permits and applying and doing all the material transfer agreements. So especially as a biobank, that's something we really need to consider. So we don't do anything underhanded or, put in socks in suitcases when you travel. But it really makes research difficult to do in South Africa, especially when you're describing new strains and the ability to deposit these in international collections. It's really difficult. So DSMZ will not take strains from South Africa because of such tight regulations. So it's both it really affects the ability to promote and do science at an international level. And also in the past, I've had international collaborators reluctant to work with us because of such strict regulations. But when you just do all the necessary paperwork, which is a pain in the backside, you can take up to 2 to 3 years to get the permit from the regulatory bodies in South Africa. So it's difficult, but doable and manageable. Yeah.
Joanne? So it's a broader question, but it just speaks to what I thought was Scott's brilliant talk. In my thinking, when I invited Scott to give that talk, was because I know you've been a brilliant mentor to many people, and particularly in the next session, Vokarea Odorin is going to be speaking, and I think your thoughtful way of how you presented the bigger societal, cultural issues around microbiome research in particular. But to get to my point, for both maybe yourself and Marla, who work in the context of South Africa and have been part of really large-scale collaborations around this issue, like I know Nikki asked about the Nagoya protocol. What I wanted to know, and the purpose of this session that Anna's headed, is the richness of the biodiversity and what a global resource that is. So I just wanted to, we often think about framing it in the context of the missing microbiomes, but it's almost like we're selfishly keeping stuff that could be this massive global resource, and how to reframe the discussion around that.
Yeah, I think it's a real challenge, because and there was a lot of debate in H3 Africa around data availability and publication policy precisely around this issue, because on the one hand, you want data to be made available to the world so that science can advance. And there are many examples where people say sequence something great and they don't have capacity to work on it, and they just sit on the data for years and years and years, and in some cases gets lost, and then science doesn't advance. So you want to avoid that. On the other hand, we know that we've got fantastic data. If I just I could put it into EGA or into ENA, and other research groups who've got more capacity than us will put 3 postdocs on it, and then they'll suction it up, and then we don't develop science on the continent. So I don't think there is an answer to that, but I think there's part of a process, and it's also about building the right collaborations. So we've got fantastic collaborators at Stanford who I think are very respectful of us and of the communities that we work in, and they help build up our capacity. But we've also got examples of people who are very transactional in their approach. In fact, maybe not transactional. They come with money and capacity. They drive something short term, they get their paper, and they're gone. And that leaves long-term consequences of lack of capacity development on the continent, and in some cases, burned relationships with communities that prevents future work. And there are examples of that have done really irreparable harm to African science.
Maybe I can give a different angle. Something that I really struggle with is wear 2 hats. As a chair, you need to do knowledge development and capacity training, but then as the director of an institute where I'm trying to push innovation, you want to do more commercial and industry-based work, and you can't publish before you've patented, and you can't patent if you have divulged anything. So anything I said today is forgotten. None of that can be patented. So there's always this constant tension, and so I don't have the answer for you. I'm just expressing a constant tension that I face. When do we collaborate and invite partners and to further the science and disseminate the science versus wanting to really make something economically viable that we desperately need in South Africa as well. And so there's this parallel position that we sit in South Africa, and I think that's why a lot of people sit on their information because of such tensions. So, sorry, no answers.
I think Margaret has a question, and then Val.
Okay. My question is to Dr. Hazelhurst, and you describe for us the collaboration by various groups that had their own projects but came together to do one project. I just want to hear the process of how you worked together, or this group worked together to use data and to produce papers. What kind of process was it, and how did you get to a place of being able to produce so many papers?
So the collaborative work is partially top down and partially bottom up, and I think that is required in any collaboration like this. So there was an incentive from the funders, is that they made some money available for collaborative work. So, for example, we realized that for much of the work in H3 Africa, we needed to have much broader genomic diversity of data available in order for the individual groups to do their work successfully. And then it made sense to come together to do that job rather than every group trying to do its own thing. And the funders were very amenable to us coming together to pitch for example, to do whole genome sequencing of 350 samples. So by coming together, that was attractive to funders to say, well, we see that this is really going to have a payoff because you'll have the data available, you can design a new genotyping array, and there'll be results coming from that. And there are other examples of collaborative work in H3 Africa that came about because funders were prepared to listen to people coming together. So you as a scientist have to say, I'd rather not be the PI on a project, but work together with someone else to do something that's going to be more meaningful. And I'll be able to bring in extra resources because funders will look favorably at seeing as something that really is meaningful. And it builds in a capacity development aspect as well.
So I have a question for Doug. Firstly, to say to all the speakers, it's been a fabulous session. Thank you. What is known about the role of phages in the control of the vaginal microbiome ecosystem?
Yeah, I think it's a really great question. And the answer is not a lot. That screen that we did of sort of these all-by-all interactions was originally designed to actually find phage interactions. So we expected that activity would be mediated by a phage. But in fact, we quickly learned that it was heat stable and protease sensitive, and heat sensitive and protease sensitive as well, so that it was a protein and it ended up being, a small protein. So that's why we sort of pivoted to kind of look at a biochemical approach to isolate that protein. But originally we thought that it would be a phage, and that's what we were looking for. So I think there's a lot more to be done. And as you know, like in many ecosystems, there's this sort of phage predation that goes on that actually helps shape community composition. And I think very likely that may be occurring in the female.
It's a very productive future area for research. Can I squeeze in a question to Scott? Because you gave a brilliant talk, Scott. Thank you so much. So thought-provoking. the misalignment between the values and the mode of collaborative science in many ways, I think, is very profound. And it's in many ways the collaborative mode is antithetical to the recognition and reward system in the academy, which valorizes the individual. So the question is, what are the lessons here and what movement can we start here? I actually think that we can lead this from Africa. What is the movement that we can start to really start recognizing people who work collaboratively and where we can recognize the contribution of teams, particularly in university systems, which are so utterly hierarchical? So maybe it's more of a comment than a question, but I think it's something I feel very strongly about. Okay. And once I get started, I'm hard to shut up.
I think what's so hard about it, it's about developing culture. We had this discussion earlier on. It's about developing culture, and that's really hard. And I've seen that being in a negative way of being an external referee for someone getting promotion at another university, and seeing it how that university was completely driven in a by point system that did not value collaboration at all. So I think the answer is it's a hard slog. It's a process of engaging with universities, with funding agencies, with NRF, so that we establish a more meaningful way of evaluating individuals. Sorry, that's the best I can do.
And before closing the session, Linda Gale has the last question.
Thanks, Anna. I'm going to be quick because I realize it is, but I've just wanted to pick up on that point. I think it's a brilliant point for us to just think about more, and appreciate your talk as well for that reason. But I'm going to sort of ground us back on day to day, and ask you, Doug, how important then, and maybe looking at the cohorts, the Seattle cohort and the Fresh cohort, how important is it intravaginal sort of cultural practices when it comes to this work? I mean, we hear it raised, we get the data, but how important actually do you think it is, perhaps?
I think it's very important. In the Fresh cohort, we have some sense that about 15% of the participants report using some sort of intravaginal drying agent. And I was just talking to Joanne about this at lunch. I don't think we really fully understand of sort of what the effect is on both sort of the microbiome and also on the host kind of inflammatory environment. But obviously, it seems like there could be a very important impact, but I think we just haven't really studied it enough to really fully understand that. But I think it's extremely important.
Again, related to that, there were 2 online attendees who were wondering whether you took diet into account when comparing the South African and US cohorts.
We have the dietary data, actually, but we haven't looked systematically at that and to see whether or not. I think that one thing that, though, I do believe is that rectal colonization may actually influence vaginal colonization, and there are some reports of doing that, but people haven't really used sort of newer metagenomic sort of approaches to sort of really look at sort of subspecies variation and whether or not there's strain sharing. So I think diet and the gut microbiome may actually really have an important effect on the vaginal microbiome.
All right. Thanks so much for everyone attending online and in person. Thanks so much to 3 amazing talks. It was really inspiring to see, leaders in different fields come together. And I think we'll meet again in half an hour for the final session of the day. Thanks so much.